This is the rest of the video for Chapter 8 on genetics. This will be DNA repair and looking at recombination strategies for prokaryotic cells. So let's pick back up where we left off. DNA, as mentioned, DNA polymerase 3, during the process of DNA replication, inherently to all DNA polymerases across all species, incorporates mismatches on a semi-regular basis. There's a very, very low rate of mismatch, like mutational incorporation, but it does happen. So DNA polymerase 3 does have built in its own repair mechanisms. So these are different ways that a cell has to repair those mismatches before they actually become integrated in that genetic sequence in that second replication round. These are base incorporation repair mechanisms. So first and foremost, DNA polymerase 3, as it's replicating DNA, can sense that bulge in the connection between these mismatched nucleotides and stop and essentially reverse, cut out the mismatch region and replace that mismatch, So it can fix or repair any mismatches as it moves forward. Sometimes it doesn't fix it, sometimes it doesn't catch it, but that is one. So one strategy of repairing DNA is by the DNA polymerase 3 itself. If it doesn't catch it, then that mismatch, so the incorrect base pairing between nucleotides that has been created, can be recognized by other enzymes that are essentially constantly seeking out and searching through the DNA to look at these these mismatches. So there's mismatch repair machinery, a whole complex of enzymes that come and they can sense that bulging, that incorrect base pairing in a new strand of DNA. And if they do sense it, they stop there, essentially cut out one of the two strands. And DNA polymerase 1 will come in, fill up that gap with DNA nucleotides, and then that ligase that we've seen before will come in and fill the gaps. It started raining again. Let's look at a picture of this in a second. Okay, so this is looking at mismatch repair up at the top, and what you see up here is methyl groups. These are methyl groups that are added on to nucleotides, so the adenine nucleotides in prokaryotic DNA. In fact, this is one of the strategies that, or one of the differences between prokaryotic and eukaryotic DNA is the methylation patterns of DNA. So this is a template strand, and you can tell because it's methylated, the new strand has not yet been methylated here. So here is an example of a mismatch that has been created from that DNA polymerase accidental incorporation of the wrong nucleotide. As mentioned, there is machinery that can sense this bulging, this mismatch in the base pairing and cut out the newly synthesized strand. It can tell which one is which because of the methylation patterns. The enzyme that does this cutting is called an endonuclease. You may have heard of this before if you're familiar with genetic engineering. We haven't talked about this yet, but we may discuss this in genetic engineering. Endonucleases are nuclease, so nucleotide cutters, enzymes that cut nucleotides internally. So these cut inside this strand of DNA at that phosphate backbone, that sugar phosphate backbone. Anyway, so it will cut out a segment of the newly synthesized strand, and DNA polymerase 1 will come in and fill that gap with the appropriate complementary nucleotides, and then DNA ligase will come and seal that gap. There's a couple other repair mechanisms. One is called photoreactivation or light repair. This is when there is UV damage. So UV induces a very particular type of damage to a double-stranded DNA molecule called thymine-thymine dimers or pyrimidine dimers. These are two thymine nucleotides that are adjacent to each other on the same strand will form covalent bonds with each other instead of their normal hydrogen bonds with their adenine complementary nucleotide. When this occurs, it also changes the conformation of that DNA strand because you have abnormal binding. And this also can be sensed by particular repair machinery. So the thymine-thymine dimer is very unique to UV damage, and there is a particular enzyme called DNA photolyase. It's an enzyme that is responsible for detecting this abnormal pyrimidine binding or these thymine-thymine dimers that are formed upon UV damage. Their job, the job of the DNA photolyase is to sense these these mutations and go and cut out that one strand of DNA that has the mutant pyrimidine binding. These are again endonucleases, so there's excision enzymes, which are endonucleases that cut out a short segment of DNA. And then, as you may guess, now you have a gap here, so DNA polymerase 1 will come in and fill up that gap, and DNA ligase will then seal that new, repaired double-stranded DNA molecule. So you have the same kind of fixer or repair machinery involved, but in this case, the one that actually does the detecting is called the DNA photolyase. Now this is when a cell has minimal UV damage, so just regular exposure to the sun or low grade exposure to UV. But when there is extensive UV damage, so the genome is peppered with these thymine-thymine dimers all over it, essentially when that damage is too great for that DNA photolyase to be effective in repairing this thing during the normal cell cycle, the cell can induce something called SOS repair. This is a special polymerase that essentially just goes and bypasses those thymine-thymine dimers. Usually this will lead to a mutant cell. Actually, always this will lead to a mutant cell, a cell that has mutated proteins. Most often, if there's extensive UV damage, that cell is going to be destroyed. It will be non-functional or non-viable cell. But on occasion, that SOS repair machinery can essentially generate a mutant cell that still has its essential functions intact and can still do basic metabolism and replication. So you may still have some a viable cell when SOS repair is induced. Remember SOS repair because when we get into our chapter on, we already talked about it. When we talked about viruses, so our chapter 5 on viruses, we talked about the virus, we talked about temperate phages, and when they enter into that prophage stage, so when that temperate phage has been incorporated in, has the genome incorporated into the host cell genome, it's called the prophage stage. And I mentioned to you that there is a repressor that represses something called the excisase for that prophage stage, and so it keeps that prophage in its indefinite prophage stage. And one of the inducers of the excision of prophage to make a lytic infection is SOS repair. So this is obviously indicative of extensive damage to the cell, and it makes evolutionary sense that when SOS repair is induced, that this prophage will excise out of the DNA and essentially jump ship, jump from a sinking ship kind of thing, try to get out of that cell before it dies, because most likely when SOS repair is induced, that means the cell has undergone such extensive damage that it likely will die. The last section here is talking about DNA recombination. So as mentioned before, a asexually reproducing cell must have strategies to increase their own genetic variation within their population to ensure survival of that species upon environmental changes. So these are 3 different strategies, evolutionary concepts that prokaryotic cells, or evolutionary, I guess you could say evolutionary strategies that prokaryotic cells have adopted to increase their own population survival in changing environments. These are all horizontal gene transfer strategies. Here's one picture that we've seen before. This is an example of conjugation, where that giant extension here from one bacterial cell to the next is that sex pilus that extends from an organism that has the ability to produce it to one that does not. And this plasmid is transferred from one to the other. So let's look at this in a little bit more detail. But the idea here is the transfer of DNA from one organism to another to create genetic variation, and sometimes can leading to a new strain identification. First, let's talk about what type of genetic material is transferred. Most of the time, we are discussing plasmid transfers. That's true for conjugation for sure, and oftentimes even transformation, so naked DNA being transferred from the environment to one cell. Oftentimes these are plasmids as well, but small chromosomal fragments are also transferred. So these are exactly what they sound like, small fragments of the chromosome, usually from phagocytosed bacteria that are lysed and there's essentially their contents are spit out into the environment, or from other environmental changes that causes lysis of the cells in the neighboring region. So think like biofilms or microbial communities in nature or in the environment, inside the body, outside the body, that there's always neighboring cells, and the environment is constantly fluctuating. So you'll always have bacteria that are living, growing, actively metabolizing, actively replicating, and ones that are dying, and oftentimes being lysed open due to host factors or due to environmental factors. So these small chromosomal fragments are generated just from lysed cells, and these fragments can sometimes be taken up through particular receptors on the surface of what we call competent cells, ones that have the ability to take up naked DNA from the environment. Or we could be talking about bacteriophage transfer of genetic information from one cell to the other. This is transduction. And during those accidental incorporations, usually they are chromosomal fragments that are taken up. And so we'll look at both of those examples. Now, two important things about these things for plasmids: these guys have an origin of replication, so the ORI site. Plasmids have their own ORI site. So this becomes important to consider because when plasmids get transferred into a new cell, a recipient cell, the plasmids have their own capacity essentially to replicate independent of the cell. They oftentimes contain beneficial genes, so genes that can confer beneficial properties to the host, but not essential genes. So usually a bacterial cell does not require a plasmid for survival, unless of course it's for an environmental change, like an antibacterial drug or something. For the small chromosomal fragments, these are fragments of chromosomes, so it's very, very unlikely that they will have their own origin of replication. Therefore, the only way that these can be propagated in the population is if they are incorporated in their recipient cell. I think I have a picture of what that looks like. So at the top here is a picture of a DNA fragment that has no incorporation, also has no origin of replication. And in that picture, you see that this bacterial cell that was a recipient of this small chromosomal fragment, this DNA fragment, it may or may not receive some benefits from that DNA fragment, but this DNA fragment is not replicated along with that bacterial cell. So whereas so you'll see in the daughter cells, because there's no incorporation of this fragment into the chromosome, and because it doesn't have its own origin of replication, it will not be replicated, and therefore that small fragment is just going to be passed on to one of the daughter cells and one of the daughter cells subsequently. So there's no incorporation, therefore this is not integrated into the population. However, when that DNA fragment does get incorporated and becomes a part of that bacterial chromosome, now this will be passed on to progeny, replicated along with that bacterial chromosome. So now this has been integrated in the population and is considered increased genetic variation. So these are the three forms of transmission or transfer of genetic information. The first is transformation, where naked DNA usually in the form of a chromosomal fragment, sometimes in the form of a plasmid, can be taken up by a competent cell in the environment. Transduction is using a bacteriophage, or when a bacteriophage accidentally incorporates a piece of a host bacterial chromosome instead of viral DNA, and it can transfer that from one host cell to a recipient cell. So we'll look at two examples of this. And then finally, conjugation using that sex villus. So let's start with transformation. I keep using this term competence. So a bacteria that is able to bind to naked DNA in the environment and bring it in through a particular receptor. This is called a competent bacterial cell. There is natural competence that occurs. That means that these guys naturally have their receptors and they can take these naked DNA fragments up from the environment. E. coli is notably is very well known for its competency. Other really great pathogens like Pseudomonas, you guys have heard of this before. We talked about Pseudomonas as one of the leading pathogens in nosocomial infections, ones that arise in the hospital setting. So one of the reasons that they are one of the more resistant organisms is because they have such a great capacity for increasing their genetic variation. They love to, they are able to take up a lot of DNA from the environment, and they're able to conjugate with many different organisms. so they can vary their genetic information, which increases their ability to survive in many different environments because they just keep gaining genetic variation. And we'll see that in just, actually, we'll talk about what those particular gene variations are when we get into our chapter on antibacterial drugs and how bacteria actually do resist these different drugs. And so you'll have a better understanding at that point. So natural competence is shown in this picture here, where there's some type of receptor that takes up a fragment of DNA. Now, another thing to note here for transformation is that almost always, and pretty much always, but there are some exceptions to the rule, this naked DNA, even if a bacteria like E. coli can take this up from the environment, if it's from a cell from a different species or a different genre altogether, like for example, if it's Staphylococcus or Lactobacillus or something from the environment, and it takes up that fragment of DNA, if there is not enough similarity between the genetic fragment that comes into the cell and that similar piece on the E. coli or the host chromosome itself, then there actually will be no incorporation. So there must be enough similarity between the incoming, essentially donor chromosomal fragment and the recipient's chromosome that there is, we call it homology, right? It doesn't mean that they're exactly the same, but there must be a high enough similarity between those fragments of DNA, the incoming chromosomal fragment and a piece that is very similar to it on the host chromosome, in order for incorporation to occur, because incorporation occurs through a process called homologous recombination. So you'll see in this picture that only one of the double-stranded DNA molecules that are in the environment enters into the cell. That fragment or that single strand of DNA will come in and it will essentially align with that same exact or very, very similar region on the host chromosome itself. And then once that fragment aligns there, there is machinery that actually will exchange, will cut out a piece of the host chromosome and stick in that, that homologous region from that fragment from the environment. So this is homologous recombination, recombining fragments of DNA of regions of homology where they're super, super similar in their nature. And this is why if a fragment of some totally unrelated species comes into a bacterial cell, there will be no incorporation, because they have to match up with very, very high degrees of similarity or homology. But once that does happen, essentially it's going to be a little bit mismatched, because there will be some variation likely from one similarly related species to another, but there'll be enough genetic variation that this can confer some benefit. So upon replication, now we'll have one of the daughter cells where that was the template strand. That daughter cell will now have a very different sequence of DNA at that site than others in that population. And this is still within the population. So now we've just increased genetic variation within that one population. So whereas that was natural competency, we in our genetic engineering lab had induced competency, or made a bacterial cell artificially competent by, in our example for genetic engineering with the P globoplasmid, we used heat shocking, right? Heat shocking was like shocking open the pores of gram-negative bacteria, those porine proteins, so that things could come in relatively easily. There are other examples of inducing artificial competency in bacterial cells for the purpose of taking up naked DNA from the environment. So if we were doing other genetic engineering experiments, this is a picture here of electroporation, where an electrical current is applied to a bacterial population to do the same thing, to essentially shock open the envelope of a cell just for a very short window of time, so that the naked DNA from the environment can be taken in. There are other ways to do this, but these are the two most common ways of artificial competence. This is I don't have the picture here. I think I took out that picture of a homologous recombination, but that's okay. Okay, so that was transformation. Transduction is with a temperate phage. Actually, sorry, generalized transduction is with a lytic phage, and specialized transduction is with a, do I have that in here? Oh, good, I have both, is with a temperate phage. So let's look at generalized transduction first. Transduction is when a bacteriophage accidentally essentially transfers genetic information from one infected bacterial cell to another infected bacterial cell. And let me just show you, we'll just walk through this image first, and if needed, I can draw this out too. Okay, so in this picture, picture number 1, a bacteriophage injects its nucleic acid into a host cell. We know this process. This is a lytic phage, so we know the steps involved in this. This is an attachment or adhesion, penetration, where the nucleic acid is injected in the cell. Now we have synthesis of, so transcription, translation, synthesis of viral parts, and replication of viral nucleic acid. And then we have spontaneous assembly and release. So oftentimes during a lytic infection, one of the strategies or one of the things that bacteriophages do is release an enzyme that essentially degrades or breaks up that host chromosome. So let's see in this picture, let's say that that phage had that enzyme and it broke up the host nucleic acid like a DNase. And during the process of, so here's synthesis and phage replication or replication of phage DNA. And here's the separated little piece of that host chromosome. Maybe there's a whole bunch of host chromosomal pieces floating around this cytoplasm. In this picture, during spontaneous assembly, whereas all of these phages spontaneous assemble and the phage DNA gets incorporated in the phage head, here is one phage that accidentally, instead of incorporating phage DNA in the nucleocapsid, it incorporates the host chromosomal fragment into the nucleocapsid. Here, the next step is lysis, of course, right? So the whole cell will lyse open. All of these new phage particles will go and infect neighboring cells. And let's just look at the process where this one accidental incorporation of that bacteria chromosomal fragment goes and infects a neighboring cell. Process will happen spontaneously. This phage still has affinity, or really, I don't know what you would, an easier way to say that, but the receptors will still naturally bind to the host cell. And the process of injection will still occur, so there's penetration. But this time, instead of that virus penetrating the cell B, the recipient cell, or the infected cell with phage DNA, this one injects bacterial chromosomal fragment from cell A, right, from the first infected cell. So now we have DNA from that donor cell, and as long as these cells, cell A and cell B, are similar enough or are related, then, in other words, as long as there is homology between this DNA fragment and the DNA in this chromosome of this recipient, then you'll have that same concept, homologous recombination, where that fragment will come and incorporate itself in the host chromosome and kick off that other piece. And then you have incorporation of genetic variation from one donor cell to one recipient cell through the accidental transmission of that fragment in that lytic phage, right? So this is generalized transduction. It's called generalized because it could be any random fragment of that chromosome from this initial donor cell, any random fragment, right? So as long as there's homology between that fragment and the recipient cell, then you will have integration of that donor DNA anywhere randomly. There is another concept called specialized transduction, and this is specialized because it's always a very, very particular fragment of DNA that is right next to the prophage integration site on a bacterial chromosome. Now, if you remember, which you probably don't, but back in chapter 5, when we were talking about prophages and the temperate phage DNA incorporation into the host cell, we said that it actually was not homologous recombination as what we were talking about here. We said this is integration. So the prophage actually enters a very, very specific site on the bacterial chromosome, any recipient bacterial cell, and it actually just opens up that chromosome. Instead of cutting out a segment, it just opens up that chromosome and integrates itself right in there. So because there's a very specific region, much like we talked about replication starting at one site on the chromosome at the origin of replication, prophage integration occurs generally at one site on that bacterial chromosome at these regions of incorporation. So because of that, again, there's kind of this defined region on that bacterial chromosome where these prophages exist. So in this concept here, this is when a prophage excises out of a bacteria that is sitting in a bacterial chromosome. And sometimes there can be accidental excision in this picture here, in picture number 2. When that prophage excises out, there can be some accidental excision or cutting of that bacterial host chromosome, right? Just a little fragment of it that may be accidentally cut out with that prophage excision, with that prophage DNA. Now, from this point, a lytic infection would ensue, so the bacterial DNA would be synthesized, so transcription, translation of virion particles or parts of a virus, spontaneous assembly, replication, of course, of prophage DNA. But here, now we have a little bit of that bacterial chromosome, and this whole fragment is going to be replicated, the whole sequence of DNA that's all stuck together. And this then, after replication and then spontaneous assembly, this whole thing will be packed into the nucleocapsids of all of these new temperate phages, the bacteriophages that are going to go and inject themselves into other cells. So then, as we mentioned, this is a lytic infection. These guys will lyse out of the cell. Now here's the next step. These guys will go and find a new host cell, hopefully a healthy one. And here is step number 1, adhesion. Step number 2, penetration, where viral DNA gets injected into the host cell. Remember, these are temperate phages, so they'll enter into a lysogenic stage, or the lysogenic phase, where that DNA then just goes and gets directly incorporated into the host cell instead of causing a lytic infection. But remember that these guys have, a little bit of that original bacterial chromosome. So here, again, it's a very, very specific region of the DNA, right? Here we have that prophage goes and lines up at that region of homology, and you have integration of both that prophage and also integration of that bacterial host chromosomal fragment. So this is one outcome where you actually have now a little bit of that original host cell's chromosome integrated into this new bacterial cell, increasing genetic variation. Another outcome is actually just a homologous recombination of that bacterial fragment part into that bacterial chromosome. So you just have a full bacterial chromosome, and then you have that prophage just circularizing and forming a small bacteriophage or prophage plasmid. In both cases, you have increased genetic variation at a very specialized region right next to that region of recombination, initially with that prophage excision. And this last form of genetic variation from one bacterial cell to the other is called conjugation. And this is using that specific structure called the sex pilus to connect two bacterial cells. And again, just as we mentioned with competency and transformation, this is usually between organisms of the same species, with some exception. Give me a second here. My son is starting up the ATV. So this is the direct bridge. Again, usually this is from the same species, but with some exception, there are some pathogens, as mentioned, Pseudomonas and E. coli, that are actually quite promiscuous, as it may be, and they really like to have bacterial sex or conjugation with members of other species. So this is definitely a virulence factor. If organisms can exchange genetic variation with members of other species, then it can drastically increase the genetic variation within that bacterial cell. So a few things, we'll write this one out. Let's start with the F factor transfer. Let me just switch to our whiteboard and I'll draw this one out for you. All right. So we are writing conjugation. That's not how you spell it. Conjugation, J. Conjugation. So let's do the F factor transfer first. Here is one bacterial cell, and this one we're going to call F positive. The F factor, right, and here's bacterial chromosome, and here's the F factor. The F factor is called the fertility plasmid. And this plasmid has on it, we call it fertility, it has on it genes encoding for a sex pilus. So this allows this organism that has the F factor to generate that sex pilus, that bridge between neighboring cells. Also importantly, this guy has its own ORI site, right? So it can replicate independent of the cell. And also note that the F factor replication is called rolling circle replication. And this allows a newly replicated plasmid, double-stranded DNA, to be generated and actually be transmitted during the process of DNA replication. So it's not the exact same replication process as we've seen before with the concept of semi-conservative replication and generating, like opening up that plasmid. Instead of having bidirectional replication, as we talked about with DNA chromosomes, in this case it's rolling circle replication, so it essentially just goes in one direction. So the process is this: F positive cells can find, will bind to or will make an attachment with an F negative cell. Okay, these guys have no, of course, no F plasmid. They still have their own bacterial chromosome in there, but they have no F plasmid. So the steps involved are as follows. Step number 1, a sex pilus is formed. Right, so this guy is synthesis of sex pilus from one cell to the next. Step number 2, the plasmid must be replicated. The F factor, or F plasmid, is replicated. So this plasmid is replicated, starting with that origin of replication. And then as this is replicated, then that replicated plasmid is transmitted over, right? So number 3, transfer of replicated plasmid to the F negative cell. Excuse me. And this goes into the F negative cell and makes it into an F positive cell. And now this guy has the capacity to go and do the same process with some other cell. Eventually that bridge will be broken and this guy can go and find a new cell. Okay, so that's what we just looked at here. Here is that, let's see if I can do this. Here is that F positive cell that has the F factor or fertility plasmid. Again, step number 1 is the formation of the bridge. Step number 2 is replication of that F factor. And step number 3 is the transfer of that F factor or F plasmid to the F negative cell. And at the end of this process, you are left with 2 F positive cells. Both have the ability to form the sex pilus, and we'll go and do this with other F negative cells, right? This is similar to HFR transfer. Let's draw this one out. In HFR transfer, the biggest difference here is that the F factor, same thing, the F plasmid. The F factor here is actually integrated in the host chromosome, making these guys HFR cells, or high frequency of recombination. So let's go and look at this one. I'll draw this one out too. Okay, this is as opposed to, we'll draw these ones. Here's an HFR positive cell. And in this case, we have the F factor integrated in, sorry, that's kind of a long picture, but here is our fertility plasmid actually integrated in the host chromosome itself, right? This is the F factor. HFR again stands for high frequency recombination. I know you guys are better than me about using text. This is called high frequency of recombination. High frequency of recombination. So these HFR positives, that means that they frequently recombine, right? And this F factor has been recombined essentially into the chromosome through homologous recombination, not exactly that, but integration into the host chromosome. Now the process here is very similar. HFR cells, HFR positive cells will find an F negative or an HFR negative cell. We'll say this is F negative. Step number 1, the same process occurs. So step number 1, sex pilus is formed. From the HFR positive cell to the F negative cell, right? Step number 2, same thing. Step number 2, the F factor is replicated. But the problem here is that because this is integrated or incorporated in the chromosome, usually the ORI site is essentially kind of right in the middle of this thing. So even though the replication is similar, where you have part of this thing replicated here, this is it's stuck in the chromosome. So you're gonna have the rest of this chromosome replicated too, and all of this stuff is then sent over to the recipient cell, right? So here's the beginning. It'll get sent over. And so as this whole chromosome is getting essentially replicated during this process here of transfer, here's that beginning part that was just replicated. And here's part of that chromosome. But this process takes far too long for the entire chromosome to be replicated in this process all the way to the beginning or the end of this whole thing. So usually, actually almost always, this bridge will be broken before the entire F factor is transferred. But there is some of that genetic information that was passed from this donor cell to this recipient cell. So here, let's write down at the bottom, F factor is replicated, but this is because this is a part of the chromosome. Only a fragment is able to be transferred before the bridge I ran out of room is broken. So I guess this would be step number 3 is the transfer of that fragment, but only a part of it is able to be transferred over. Now that's kind of okay as long as you're understanding the purpose of this is to increase genetic variation. So here's that host chromosome, and even though you didn't get the whole fragment transferred over, this still will provide increased genetic variation. Okay, it still will leave this as an F negative cell, but it has increased genetic variation. Let's go and look at this. The picture, of course, from your textbook is much prettier than mine. So here's our donor cell that has that F factor integrated in the chromosome itself. This has the ability to make the pilus because that F factor still is the same thing and encodes for a sex pilus. So here we have the step number 1, that pilus is formed between an HFR negative and an F negative bacterial cell. Here is that rolling circle replication, right? And only part of that F factor, because the origin of replication is generally kind of in the middle of this thing as it's integrated in the chromosome. So a part of that will be replicated, and sort of as this is going along, that chromosome with that F factor is passed over to the recipient cell, but usually that bridge is broken far before the entire thing can be passed over. So these are considered just donated genes instead of that HFR positive. So usually the fragment is integrated, but usually this still leaves that donor cell F negative, right? It does not have the entire plasmid incorporated, and so this is just a recombined cell. It has gained genetic variation. When we get into our chapter on antibacterial drugs, we'll revisit this concept, and instead of calling this an F plasmid, I'm going to call it an R factor, because if this F plasmid has on it genes that allow for drug resistance, then this is called a resistance factor. And the same process occurs though. So we'll revisit this in chapter 10, I think. Go ahead and watch this video from your chapter textbook. This just shows you these different variations. I think this shows you conjugation. So again, these are the three formations of recombination, three forms of recombination. Conjugation that we just looked at with the transfer of that F plasmid, transduction using a bacteriophage, that's either specialized transduction with a temperate phage or generalized transduction with a lytic phage, and then transformation is the transmission of naked bacterial chromosomal fragments usually from lysed bacterial cells in the environment taken up by competent cells. With this, that ends our chapter on genetics, and don't forget this stuff. We are going to revisit it in every chapter as we get through the rest of the semester. I think that's it. Ciao.